Hello everyone and welcome to our module on embryogenesis. The first step in the process of generating a human embryo is fertilization. In fertilization, a haploid mature spermatozoon from the male fuses with a haploid ovum from the female, and this forms a zygote. Now, one thing they're going to want you to know for your step 1 exam is the number of chromosomes and chromatids present in all these structures involved in fertilization. So let's make that very clear now. So eggs and sperms are 1N, 1C. So what do I mean by that? By 1N, I mean they have one copy of each chromosome. So for example, if we were talking about chromosome 6, there is a single copy in each haploid spermatozoon. There's also a single copy in each egg. In addition, there's only one chromatid for each chromosome in a sperm or in an egg cell. Now, once the sperm and the egg merge, we now have a situation where we have 2N and 2C inside of one cell. So for example, we have two copies of chromosome 6 now in the zygote, and each copy of chromosome 6 has one chromatid, thus there is 2N, 2C in the zygote. Things get a little more confusing once the zygote forms, because one of the first things that happens inside the zygote is that DNA synthesis begins. Initially within the zygote, the maternal and paternal DNA are separated from one another inside of cellular structures called a pronucleus. A picture of this is shown on the screen here. We've got a male pronucleus with the father's DNA and a female pronucleus with the mother's DNA. Now the zygote, as we talked about in the last slide, is initially 2N, 2C. However, when DNA synthesis occurs, chromatids will be synthesized such that the zygote will then become 2N, 4C. And I've drawn a picture to help you understand this on the screen. So initially, we've got the zygote, which is the blue circle here. It's got two pronuclei inside it, which are the two green circles. And inside each pronuclei, we have a copy of DNA. So for example, let's suppose the red line is chromosome 6 from the mother and the blue line is chromosome 6 from the father. We then undergo DNA synthesis and we synthesize two sister chromatids for each chromosome. So we now still have two copies of chromosome 6. This cell in the middle of the screen is still 2N, but it is now 4C because there are four chromatids. Then the cell can undergo division and become two cells, each with one copy of DNA from the mother and one from the father. So when this occurs, the zygote divides and it now becomes two cells that are 2N, 2C. And this process by which the zygote divides is called cleavage. It's one of the first important steps in formation of a multicellular embryo. Shown on this slide is an illustration that you can find in many textbooks showing all the steps from ovulation through fertilization through implantation of the developing embryo in the wall of the uterus. So first of all, you should know that fertilization takes place in the fallopian tubes, and it usually takes about 2 days after fertilization before that first cleavage event occurs and the developing embryo goes from the one-cell stage to the two-cell stage. After the two-cell stage, the developing embryo continues to undergo cellular division and it forms a structure called a morula. A morula means ball of cells, and this is what we call the embryo when it's in the stage where it's just a clump of cells stuck together. After morulation, the next step in development is called blastulation. This step involves formation of a structure called a blastula from the morula. The difference between a blastula and a morula is that the blastula contains a fluid-filled cavity called a blastocele. In humans, the blastula is called a blastocyst, and the reason it's called a blastocyst is because the fluid-filled cavity is not in the center of the structure. The blastocyst is made up of a group of outer cells shown in purple in my cartoon here. These are called trophoblast cells. These cells are polarized, one side is different from another, and they secrete the watery fluid that fills up the blastocele, shown in purple in my drawing here. The other part of the blastocyst is called the inner cell mass. This is an apolar collection of cells. In other words, one side is no different from the other. And as we'll see, these are the key cells that give rise to all the tissues of the body. And these are the cells from which you can derive embryonic stem cells, which are sometimes used for research purposes. And here's a micrograph showing you what the blastocyst looks like, just so you know this is an actual structure that you can see under the microscope. So the blastocyst is the structure that implants in the uterus. This usually occurs about 6 to 10 days after fertilization. And once the blastocyst has implanted, the placenta begins to grow. And as I talk about in another video, the placenta is what synthesizes and secretes beta-HCG, which is used to detect pregnancy. So the key point here, which has clinical relevance, is that you can't detect a pregnancy using beta-HCG until about 6 to 10 days after fertilization, because that is when the blastocyst has implanted in the wall of the uterus. So now we've talked about morulation, where the morula was formed, and blastulation, where the blastocyst was formed. The next step in embryogenesis is called gastrulation, and in this step the blastula is converted into a three-layer structure called a gastrula, and the three layers of the gastrula are called the three germ layers, and their names are the ectoderm, mesoderm, and endoderm. And these three germ layers will develop into all the organs and structures in the embryo that will persist into adulthood. The way that gastrulation occurs is that inner cell mass, which I showed you a few slides ago in the blastocyst, forms a structure called the bilaminar disc. The bilaminar disc contains two cell layers, that's why it's called bilaminar, separated by a basement membrane. And the two cell layers are called the epiblast and the hypoblast, and they're both shown in my picture on the screen here. And I've bolded the word epiblast because the epiblast is what will develop into the embryo. The hypoblast develops into the yolk sac and some other important structures in the womb, but it does not develop into any future embryonic structures. Another important step in the process of gastrulation is formation of the primitive streak. The primitive streak is a structure that's formed by the invagination of epiblast cells. Basically, that layer of epiblast cells invaginates like I'm drawing here, and that creates a streak. This is a visible line in the blastocyst that you can see under the microscope. This is an example of a primitive streak shown on the screen here. And what you want to know in terms of step one about the primitive streak is once that is visible, this means that the process of gastrulation has started and the germ layers are beginning to form. So as I mentioned before, the epiblast develops into the three germ layers, the ectoderm, the endoderm, and the mesoderm. This is shown in my cartoon on the screen here. So what I'm circling with my pen is what used to be the bilaminar disc. It's now formed three layers. The pink layer on top is the ectoderm, the brown layer on the bottom is endoderm, and the mesoderm is in the middle. I've also shown an invagination here, which is the primitive streak. And remember that once you see the primitive streak, that means that gastrulation has begun and the germ layers are beginning to form. And then the three germ layers go on to develop all the parts of the embryo. And in a separate video, I talk about the germ layers in detail. But just some basic points here. You can basically think of the endoderm as forming the digestive system plus a number of other structures that all bud off from the digestive system. You can think of your ectoderm as leading to development of your external body structures like the hair, nails, and skin. The only exception to that is the nervous system. And to remember this, you have to know that the nervous system and the neural tube form from an invagination of the ectoderm. And then finally, the mesoderm forms everything in the middle of the GI tract and the skin. So this includes things like the circulatory system and especially the skeletal system and the muscular system. And that concludes our video on embryogenesis.